development.
Sorry.
It's okay. Sometimes they take things. I mean, same like for your glad view stuff. You said that took a while too for you. It's okay. Yeah. All right, I'm just gonna start up. Hello, everybody. Thanks for joining the online seminar series of the International EPR Society. I'm excited for this talk today because this is going to be, it's more biomedical, obviously. It's like biomolecules on DIA, on proteins. But now the problem for EPR is that we're in a frozen state, right? Frozen state is a little bit tricky if you want to check out like enzyme kinetics, folding events. So they're very smart people. I mean, beyond belief, have come up with the idea of a rapid freeze burn so that we actually freeze faster than you can actually take information or interact, and then combine it with a rapid mixing event. And once a protein mixes together, we can actually stop the time. I mean, this is like the power of EPR, right? I mean, we can stop the time in freezing and look at individual events. Now, recently, always recently, like half a year ago, I think there was a paper by Tobias Hett and Olaf Schiemann came out, where they introduced a new mixer and a new system and showed that really this system can be developed by stopping the time after only 80 microseconds. Now think about it, what you can do with 80 microseconds, all those different biological systems you can encounter. And so we decided that maybe it's worthwhile for Tobias to just give a talk and guide us through what he's really doing. And hopefully the whole method will become popular and open up new gates for us to explore and compete with other methods. So right now, I'm just going to give the stage to Tobias. And I think Tobias has a lot of things to say from Bonn under Olaf Schiemann. So please go ahead, Tobias.
Yeah, hello, everybody. And thank you, Thomas, for the kind introduction and especially the invitation to this talk. As already follows from the title, I will today show you a method that we can use to follow conformational changes in proteins. And we want to follow them with resolution in space and time. Okay, let us start. So biomolecules in general are not static, but they are dynamics, meaning they can undergo structural changes. And basically life is based on these structural changes. So what you can see here on the left-hand side is the apostate of a protein. This is a so-called cyclic nucleotide binding domain, or as we call it, CNBD. And when it binds cyclic adenosine monophosphate, CAMP, it will undergo a structural change, which you can see here. So what basically happens is that the alpha helix, this one, the C-terminal alpha helix, makes a sliding movement, so a displacement to the left-hand side. And also over here in this helix bundle, there are some changes. So the structures of the apostate and the holostate are both known from NMR spectroscopy. And what we wanted to do now is to find out how we can actually get our system from the apostate to the holostate. So what happens in between and how exactly the structural change happens. And we tackled this question using EPR spectroscopy and freeze-quenching. So for EPR spectroscopy, or for pulsed electron-electron double resonance in particular, we need a paramagnetic system. And the protein as such is often diamagnetic, yes. So it doesn't have any unpaired electrons. And in order to introduce unpaired electrons into our system, we use the spin labeling technique with MTSL, the MTSL spin label. This is what you can see here. So we attached two labels to our protein, and then we performed the PELDOR experiment. I guess most of you will know the PELDOR experiment. What you end up with is an oscillating time trace that encodes the dipolar interaction between the spin labels, from which we can infer the distance. When you analyze your time trace, you get a distance distribution, meaning the probability density of a distance between your spin labels. And from this distance distribution, you can draw conclusions on the conformation that your protein has under a certain circumstance, so either without ligand or with ligand. So the first task we had to solve was to find suitable labeling sites on our protein. And finding the labeling positions can be quite easily done using so-called difference distance maps. So what this map shows is the change in the distance from the apostate to the holostate when looking at a certain pair of amino acids that is labeled. So the distance change in the protein is color-coded in this map, meaning the bluish colors represent a rather small distance change, whereas the reddish colors represent a large distance change. And now from this difference distance map, one can pick pairs of amino acids that we label and then measure PELDOR on it. So from the map, we basically picked two pairs of amino acids that we labeled. The first one is the E289, I340 residues. In this case, we are located down here in the beta sheet, and the other one is in the alpha helix, which is supposed to do the sliding movement. And from the difference distance map, we could find a distance change of 1.8 angstroms, so a comparatively large change in the distance. The second construct that we chose is the R254E336. It's located here on the distance map. And once again, our labels are in the beta sheet down here, which is invariant during the structural change, and on the alpha helix over here. So in this case, we're at the very top, that is at the starting point of the alpha helix, and here we're a bit further down. So these are the two constructs we used for our project. So having decided on the constructs, we first of all took a look at the apo and the holostates. What we see here is the PELDOR time trace and the distance distribution that we get for the construct E289 I340. And as we add cyclic adenosine monophosphate, we see that both the time trace and the distance distribution change. So over here, we have most probable distance of about 4 nanometers. When we add CAMP, the distance distribution peaks at about 2 nanometers. So this corresponds to a distance change of about 18 angstrom. The second construct we also had to look at it in the PELDOR experiment. The apostate has a most probable distance of about, let's say, 3.8. And when you add CAMP, we get a distance change which is much smaller. So it's only about 3 angstrom. And as we can see in this case, the distance distributions are well separated from each other, so they hardly overlap. Whereas in this case, there's quite a strong overlap of the distance distributions. We will see what implications that has for data analysis later on. So now we have the constructs. The next thing we should think about is how we can actually get the time resolution to find what is in between the extreme states, so in between apo and holo. So how can we observe proteins in motion? We would like to do this in analogy to Edward Muybridge, who already back in the year 1878, so about 140 years ago, monitored the ride of a horse. And what he did was to place a lot of cameras in a row to capture snapshots of this riding movement of the horse. And when then attaching all the snapshots that we see here, one after another, he could reconstruct the movement of the horse, like in a movie. And this is basically the principle we want to apply. So we want to take snapshots of the conformational change of our protein and then see what it looks like in between. So how can we take the snapshots of our protein? We do this using the so-called Microsecond Freeze-Hyperguenching technique, so a variation of freeze-quench. In freeze-quench, what you basically do is that you have your protein and the ligand in 2 separate containers, meaning, for example, syringes, and then you quickly mix them in a micromixer. So mixing occurs in our case on a microsecond time scale. And after mixing, in order to stop the reaction, so the reaction between protein and ligands, we eject them into a cryomedium. That can either be, for example, an isopentane cold bath or metal surfaces which are cooled down to liquid nitrogen temperatures. Here on the right-hand side, you see a sketch of our device. It basically consists of a cold plate. So this thing over here is a hollow metal cylinder that one can set into rotation. Around this setup, we have a vacuum hood so that we can work under reduced pressure. Here's a mixer. So basically this thing here, in which the protein and the ligand are mixed. And over here we have pumps and containers to transfer the reactants into the mixer. We will later on see how the samples are actually prepared. So what is special about our setup is that we can use it in a time range between 80 microseconds and roughly 700 microseconds. So we work on a microsecond time scale, whereas most of the commercially available devices work on a millisecond time scale. So we are faster by a factor of 1,000. And per time point, we roughly need about 100 microliter of the protein at a concentration of 50 micromolar, so pretty normal PELDOR conditions. So this is our setup. Let us now look like how we can set the aging time, meaning the reaction time of the protein and the ligand. So the first part of the entire aging time is the mixing time. This is what you see here. So this is the micromixer of our MHQ setup. We see down here, this is the mixing chamber. We have two channels, so channel A and channel B, where the protein and the ligand are fed into. Then these two channels are again split up in further two channels, such that we in the end have 4 channels, which are then joined here in this 4-jet mixer. So over here in the middle, there the 4 channels are joined. On top of this mixing chamber, we have an orifice. This is this metal plate, which in its middle has a hole with a diameter of 20 micrometer. And by pushing the reactants through this orifice, we get a well-defined thin jet of just 20 micrometers. And the residence time of our solutions in this mixer amounts to about 1 microsecond. So the mixing time is 1 microsecond. So what comes then after mixing is the transport. So the transport time, meaning when the jet of our solution leaves the mixer over here at the output, it then travels through the evacuated vacuum chamber onto the cold plate. And depending on the length of this jet, which can be between 3 millimeters and 72 millimeters, it takes differently long before it hits the cold plate. And this allows us to set a transport time between about 40 to 650 microseconds. And the third part of the total aging time is the freezing time, which is fixed to 40 microseconds. I should add that the mixing time and the freezing time have been determined by Sherepanov and De Vries, so the guys from Delft who once invented this MHQ device. And yeah, T-mix and T-freeze are fixed, T-transport can be varied. And the total aging time of our sample is then simply the sum of T-mix, T-transport, and T-freeze. Okay, let us now have a look at how the samples are actually prepared. So here we once again have the sketch of our setup. And what we do to prepare samples is to firstly cool down the cold plate by pouring liquid nitrogen into it, then letting it sit for roughly 20 minutes, such that it is cooled down to 77 Kelvin. Afterwards, we then quickly remove the liquid nitrogen and then put the vacuum hood on top of it. What we next do is to suck vacuum until we are at a pressure of roughly 30 millibars, because then there's less air in it, so the jet which will be produced from our mixer has less of an air resistance. So it's better defined and it is not falsified, if you want to like it like that. What we then do is completely controlled by computer, is to inject the protein and the ligand into the micromixer, which then moves vertically down into the cold plate. Then the jet, so the mixture of the protein and the ligand, is sprayed onto the cold plate. Depending on the length of this jet, we have the possibility to vary the aging time of our sample. Now I have a small video in which we can see how the spraying procedure actually works. So now we will see that the cold plate starts rotating. We see a bit of liquid nitrogen flowing around there. And then the mixer moves down into the cold plate, spraying the jet, as we can see now, onto the plate. And as the spraying procedure has finished, we can again reventilate the vacuum hood and then pour liquid nitrogen into the cold plate so that our reaction is terminated, that our sample cannot warm up anymore. Then it is fixed. Afterwards, we put the cold plate into slow rotation. We scratch the sample from the surface of the cold plate and then put it into an EPR tube, compress it there such that we can do the measurement afterwards. So this is basically the procedure of preparing the MHQ samples. Okay, so as we had the setup, we first of all thought, okay, maybe we should calibrate it to see that it really works reliably and that it can also reproduce data which has already been published in the literature. And we did this calibration with the well-known reaction between myoglobin and sodium azide. I guess most of you will be familiar with it. So as myoglobin is in its apo state, we have a high-spin complex. So iron 3 is in the high-spin state, which gives rise to an axial CW EPR spectrum. This is what we see here. It's an X-band. So if we now add sodium azide, the spin state changes as we have a binding of the azide ion here and displacement of the H2O ligand, such that we get an orthorhombic EPR spectrum. As we can see here, they are markedly different. And from the peaks, so the peak amplitudes in these spectra, we can deduce the percentages of high spin state and low spin state in a freeze-quench sample at a defined aging time. And this is what we did for calibrating the MHQ setup. Here we see the results from this calibration series. We prepared MHQ samples at different aging times, so from 82 microseconds to 668 microseconds. And what we can clearly see from the EPR spectra is that the longer we let the samples react, the lower the amount of the high spin state, so the smaller this signal over here is, and the larger the low spin state signals over here become. So from the peaks in the spectra, we got the percentages of the apo state, so of high spin and hollow state, low spin, which we can see over here in this plot. We then performed regression on it, so we fitted a biphasic 2D first order kinetics to it in order to compare to literature. And one sees the fits are quite nice, and the rate constants which we got from our fits are in coincidence with the literature data that had been published by Sharapanov and de Vries, so the people who invented the MHQ device. And from this data, we then concluded, okay, our device works reliably so that we can go on with the preparation of samples of the CMBD. This is what I will show you next. So time resolved PELDOR. What we did was to first of all prepare MHQ samples of the apo state and the hollow state of the construct which had a large distance change. So now we once again have the extreme states, and now let us have a look at what is actually in between. So what happens if we prepare samples in between? what we can clearly see is that the longer we let it react, the lower the apo peak, so this one over here is, and the greater the holo peak, so this one over here is. So we see a change in the population of the apo state and the holo state. So a gradual decrease of the apo population and an increase of the holo population. We also prepared MHQ PELDOR samples for the second construct. And here the picture is basically the same, even though it is a bit more difficult to see, but we see a gradual decrease of the apo state and a concomitant increase of the holo state. So if we say, okay, we want to monitor the movements or the flipping of this alpha helix, we would actually expect a shift of the most probable distance along the distance axis. So we would think, okay, we start at 4 nanometers of the apo state, then go to maybe 3.5, 3.2, 2.5, 2 nanometers. So see a change along the distance axis. This is what we obviously do not see here. We see a mixture of the apo state and the holo state to different extents at the different aging times. So in order to further look into that and to also quantify the percentages of the apo state and the holo state, we had to find a method to do that. And quantifying the percentages of apo state and holo state can be done by deconvolution of the PELDOR traces. So what we started with was the trace of pure apo state, pure holo state, and the MHQ shot trace at a certain aging time. Now, as we can see here from the original data, is that the modulation depth is always slightly different. I mean, you may know that it depends on how perfectly you can set up the PELDOR experiment. It also depends on the labeling efficiency of the particular protein batch. So for doing a good deconvolution of the PELDOR traces, we first of all equalized the modulation depth computationally. So meaning we did modulation depth scaling. So over here, the modulation depth identical. The next step afterwards was to do the deconvolution of the traces, meaning what we did was to represent the MHQ trace, so the vector of the data which you see here, as a function of a sum of the apo trace and the holo trace. So again, a vector and a vector. And these two are the scalars, which then weigh the vectors of apo and holo. So we want to describe the MHQ trace as a weighted sum of the apo trace and the holo trace. This worked actually quite nicely. We can see that the experimental trace and the trace which we can mix in silico by mixing apo and holo overlapped quite well. And the algorithm tells us, okay, this MHQ trace consists of 28% of the apo state and 72% of the holo state. That this deconvolution works actually quite nicely can also be seen from the residual. So the residual between the experimental data and the in silico mixed data is just noise. So this is the algorithm or the procedure that I programmed in MATLAB to get the percentages of apo and holo at each time point. So we did this deconvolution for all of the time points that we acquired with MHQ PELDOR. And here's what then came out with that. So as we could also see from the distance distributions, we have a gradual decrease in the apo state and a concomitant increase in the holo state. When you then fit a Q default state rate very constant to it, we see that we get a rate constant of about 3,400 reciprocal seconds from it. For the second construct, picture is basically the same. We're also in the same order of magnitude, which is on the one hand quite reassuring because it's some kind of reproduction. And it also implies that the kinetics, which we observe here, is independent from the labeling side. So having seen that we have a gradual shift in the populations from apo to holo, we wondered, okay, what is it actually that we monitor here? So it's obviously not the switching of the alpha helix. And then we thought, okay, maybe is it just ligand binding? And in order to test for that, we performed saturation experiments. So in order to find out whether MHQ PELDOR monitors ligand binding, we produced MHQ PELDOR samples at a fixed protein concentration of 150 micromolar and at a fixed aging time, then varying the concentration of the ligand, so varying the CMP concentrations. What we can see from the distance distributions is that, of course, the larger the CMP concentration is, the less of the apostate we get and the more of the holostate we get. This is intuitive. But what we can also see is that as soon as we are at a concentration of 10 millimolar of CMP, we are in saturation. So exceeding a concentration of 10 millimolar doesn't lead to any change in the population of apo and holo anymore. And this is what we also see here from this plot. So when we are at a ratio of CMP to protein of about 66, we're in saturation. So no more change in the population of apo and holo. So we can conclude saturation was already achieved at a ratio of CMP to protein of 66. So as our time-resolved studies that I showed you on the slide before was conducted at 100-fold excess. So at this point, we could conclude that ligand binding is actually below the time resolution of MHQ. So in the MHQ PELDOR experiments, we did not observe the ligand binding events. So the question is still, what do we see here? And in order to find that out, we took a look at the reaction using in silico methods, so meaning molecular dynamic simulations. So first of all, we did was to run an MD simulation on the apo state as we have it in the PDB, such that we have an MD trajectory and know how the protein behaves and what it looks like in the computer. The next step was to do the same with the holo state, also with the structure as it has been deposited in the PDB. Now afterwards, as we have apo and holo in the computer, we wanted to simulate the conformational change itself. So what we did was to model the CMP ligand into the binding pocket of the apo state. So modeling it over here in the position as it is also found in the holo state. And then we let the MD run in order to see what the time scale actually is. So how long it takes to transfer this simulation construct into the holo state. Here we can see the results that we get from the MD simulations. So we have plotted the distance between the C alpha atoms of the amino acids that we also labeled for the PELDOR experiment and monitor them as a function of the simulation time. So we simulated about up to 3.5 microseconds. And we can see that the apo state is pretty well defined at a distance of about 3.5 nanometers. We also did it for the holo state, which is located down here at a distance of about 2 nanometers. So now the interesting part comes, what does the simulation of the conformational change look like? And this is what we see here. With the zoom in on the right-hand side. So here, this is the zoom in on the first 30 nanoseconds over here. So what we see, what happens is if we simulate the conformational change, is that we start at the level of the apo state over here. Then we have an extremely fast switch over here. So it's about 7 nanoseconds before we end up in such an intermediate region, before then again switch down to the level of the holo state again on a nanosecond timescale. So MD basically shows us that the conformational changes happen on a low nanosecond timescale, which is way faster than what we could resolve with the MHQ device. We could also saw from the ensemble of 25 trajectories that we computed is that the trajectories do not look the same, meaning it takes differently long before the conformational change actually occurs. And the so-called waiting time maybe between a couple of nanoseconds up to several microseconds. However, the flipping itself, so this conformational change, was always on the order of a few nanoseconds. Now, having on the one hand side the experimental data, which show us a change on the microsecond timescale, and the MD data, which show us a change on the nanosecond timescale, well, we had to find like a mechanistic model in order to interpret the data. And this is what we can see here. Our interpretation is in the framework of so-called dwell times or waiting times. This is a free energy profile that we created for our reaction. And where we start is the protein, free protein in the apo state. So we have the apoprotein and the ligand separated from each other. Then we have the first free energy barrier over here, the ligand binding, which occurs on a nanosecond timescale, or at least below the MHQ time resolution, as we saw from the saturation experiments, before we then end up in a so-called apo-ligand complex. And this apo-ligand complex is a structure of the protein where it is still in the apo state, but the ligand is already attached to the protein. Then the second free energy barrier, which we have over here, is the conformational change, which occurs on a low nanosecond timescale, as we could see from the MD simulations, before we finally end up in the hollow state, so with the ligand tightly bound into the binding point. And what is in between here, in this apo-ligand complex, we have the so-called dwell time. So this apo-ligand complex exists for a certain lifetime, in that it accumulates the thermal energy that it needs to overcome this second free energy barrier. So it takes a certain time before it can jump over this barrier. And this lifetime is on a microsecond time scale. This is what we could observe using the MHQ. So we neither saw the ligand binding, nor did we see the conformational change, but the lifetime of this apo-ligand complex. With that, I would like to conclude and thank all people who contributed to this project. First of all, my PI Olaf Schiemann for giving me the opportunity to work on this interesting topic, and also for the constant support throughout all the time we worked on it. Then it is a collaboration with our partners from the Center of Advanced European Studies and Research here in Bonn, Tobias Hett, Reinhard Seifert, and Benjamin Kalb, for doing the biochemistry side of the project and for the MHQ experiments. Then we have a collaboration with the Max Planck Institute for Biophysical Chemistry in Göttingen with Reinhard Clement and Helmut Kruppmüller, who quite nicely introduced me to the world of MD simulations and gave me the opportunity to run the MDs on their cluster. And of course, we acknowledge the DFG for funding of the project. Here on the right-hand side, you can see a snapshot of our paper that came out in April this year. And with that, I would also like to thank you for listening, and I'm happy to answer your questions.
Great, thank you, Tobias. I think you didn't fail to amaze, as I can see that Ilya already has a question. But the good thing as a moderator, I can ask my first question. So that's best. Hey, this is really cool, and I like it. Now, in regards to the dwell time, I see where you came from on the MD simulation. But do you think there's just an intermediate? Because if one looks at the binding curves, what you showed now, given it fits relatively well, but one can easily think that that dwell time is moderated by a very short-lived intermediate, which could, as you see, if you add them up, there's going to be some deviation in there. You can also see that the residual, they're not perfectly matched. There's something in there. Of course, we will not know and might not be observed, but what do you think about that? I mean, that's like the holy grail of freeze-quench. You look for intermediates.
I mean, you may be totally right. We have this intermediate lifetime over here. There's definitely a different distance. And yeah, to a small extent, there may be this contribution in here, which may, as you already said, be the reason for this residual that we have. So it may not only be the noise in the trace, but also some kind of intermediate.
Great, awesome.
I would say the major contributions are the aqueous state and the hollow state.
Yeah, of course, but that's cool. I mean, this is great. Ilya was the first one to ask the question. Ilya, do you want to just ask it in person, or do you want me to go ahead?
It's a pretty simple question. This is the best, the most consistent change with the integrals of the distance distribution. You know, one peak consistently goes down, the other consistently goes up. Was there any magic involved in the data processing? So I'm just curious about the data processing method there, because that's probably the highest quality data I've ever seen for kinetic studies of this kind.
Yeah, thank you for the question. What we did was to do background correction with the analysis. And then, as I showed, we did this deconvolution. So we plugged in the background corrected time traces into the MATLAB script that I created for doing the modulation depth scaling according to literature. And then it's just a solution of this linear equation that we did. And what we also tested was to see what the influence of background correction is. So whether it makes a difference if we use the background corrected traces for deconvolution or whether the raw traces are used. And the outcome is basically the same. So it doesn't matter if we use the processed traces or the raw traces.
Great, thanks, Tobias. Eric, I think you were the next one, and then John.
Okay, well, I have, so if I understood what you were saying to Thomas, you think that you might see the apo-ligand state, but it would be relatively small and in these residuals here?
Mm-hmm.
Yeah, okay. It wouldn't be a significantly larger component of your distributions?
Yeah, exactly. It would just be a minor contribution to the distribution.
The other question I have, which is sort of from out of left field, I know there's been a lot of work on the effects of high pressure on proteins structures. And I'm wondering if there's any reason to think that going to this low pressure environment might affect the structure, or shift the equilibrium in some way.
Mm-hmm. Yeah, you're certainly right. That may in some way affect the structure. But actually, in terms of the preparation of the MHQ PELDOR samples, it's the only way that you can get sure that the aging times are correct. So, I mean, if there is some impact, it would certainly be low, as we can reproduce in silico. So, for example, with MTSSL Wizard or MMM, or distance distribution. So I would say there's not a drastic effect of the low pressure on it.
All right. How about Jan? Would you like to ask?
Hey, sure. Hi, it was very nice talk. Thanks. I liked it a lot. And I had a few questions actually. One very practical was when I was in the lab of Song Han. We tried a little bit this with this flash freezing quenching. And I always had an issue with the scratching of the protein when it's frozen, and how much waste here, and if you condensate some water from the air, etc. I'm just wondering what you do for that, and if that is straightforward in your setup, or if there's a little bit of an expert hand to do that.
I mean, you're completely right. So how we do the scratching and freezing is basically that we put the cold plate into slow rotation, then scratch it with a scraper from the cold plate. And then for packing the samples into APR tubes, we have constructed such a double-walled funnel. So in the outer part of the funnel, you can cool it with liquid nitrogen. And the inner part of the funnel, there you can put in the scratched EPR sample with some nitrogen into it so that it stays cooled. And then we have this EPR tube attached to it such that you can pack it with a rod. And I completely agree with you that the condensation of air moisture is an issue. It leads to a dilution of the spins in your sample. So far, the only thing we can do is to pack it as quickly as possible. So to stay it for the least amount of time on the air, such that, yeah.
Do you spin it down a little bit? Do you spin it down to help going down or no?
No,
no,
we
okay.
just let the liquid nitrogen evaporate and then pack it. It's actually quite crucial to pack it really densely, because if there is liquid nitrogen trapped in your sample, I mean, it may get shot out of the tube afterwards. So you have to really compress it a bit. And it is also good to compress it, because you just have a very tiny amount of sample in your resonator. So it's crucial to compress it.
Okay. If I may, can I ask one more question? Also, you mentioned this 40 microsecond freezing time. And I assume this is very dependent on the quality of your jets, meaning the size of your micro droplets that hit your frozen plate. And I guess there is also a lot of science here in mass spectrometry and even in those free electron X-ray lasers. I'm just wondering, do you control that? Is that something where you developed a lot, and how do you know that you are at this 40 microsecond based on your jet system?
So in order to make sure that our setup works reliably and that we indeed reach the aging times that we think we reach, we did this calibration. And there are further details in this paper from Sharapanov and the Freeze. They did some measurements on the jet speed and so on. So the jet is quite fast, about 200 meters per second. And there's some more literature on that, but we didn't go further into that in detail.
Yeah, okay, just and so my last tiny question was about the when you deconvolve in the population. You just answered a question about that. Did you try, just for curiosity, to integrate your P of R and compare, just out of curiosity, how this compared to when you fit the actual dipole evolution time, and see if there is, how far you are in agreement between the distance domain and the time domain?
Yeah, of course, we also did that, especially for this construct. It works quite nicely. So we have apo and holo well separated. You can indeed integrate it, and the result's the same as we got from the deconvolution. For this construct, I would say deconvolution is the only way that one could use to really get the percentages of apo and holo, because integration is with those overlapping distance distributions actually impossible.
But otherwise, you're saying you're quantitatively exactly the same, or almost exactly the same, between the convolution and the integration from the first.
Exactly. It was the same for the convolution and integration.
Okay. Cool. Thanks. That's it for me.
Thank you, John. Hey, Hassan, I think you have a burning question. Why don't you go ahead?
Yeah. Very, very nice talk. So I have the very, very quick question is related to what he's showing right now. So are these shoulders that you are saying in this distance distribution, can you comment on this, or do you think they are real? It looks like they are real, because by looking to the short distance, definitely you do have multiple frequencies. So is this related to other conformations, or? Thank you.
I would